Hello everyone. Today we're doing part 2 of our response to Long Story Short's rebuttal to his video on whale evolution. So let's jump right in.
Next, let's take a look at Archaeopteryx. They say I've got a bad quote there. In the original it says, the small Silorosaurian dinosaurs related to Archaeopteryx. And I have that in brackets theropods. Okay, is this a bad quote? Are the small Silorosaurian dinosaurs related to Archaeopteryx theropods? Yes, they are. They're a subgroup. There's nothing wrong with the quote or how I used it.
Wait, hold on a minute. Silorosaurs are theropods. They're a subgroup. Therefore, there is nothing wrong with the quote that equates theropods in place of Silorosaurs, saying that they appeared after Archaeopteryx? Is he joking? Apparently, he just isn't familiar with the fallacy of division and composition. What's true for the whole isn't necessarily true for the parts and vice versa. What is true for Silorosaurs isn't necessarily true for theropods as a whole. The first theropods appeared in the Triassic, long before the appearance of Archaeopteryx, a fact that has been known for well over a century. The first Silorosaurs date back shortly before Archaeopteryx, which is the actual point of contention that was discussed by Robert Martin in his book from which the quote originated. So these are not interchangeable whatsoever. LSS saying that they are interchangeable on the excuse that one is a subgroup of the other just boggles the mind. Imagine if I gave a quote that says, the first humans arrived in the Americas during the 15th century. But the original quote actually says the first Europeans. Would LSS come to my defense saying that since Europeans are humans, they are a subgroup, there is nothing wrong with my quote? I really hope not.
Okay, well what about the more recent fossil finds that Jackson brings up? The paper he cites notably confirms the long-known temporal paradox with bird evolution. Yet, Archaeopteryx was still trumpeted for years previously in spite of this known difficulty.
Yes, Archaeopteryx was considered a very significant transitional fossil for a long time, even before this gap in the fossil record was filled. And that's okay because a gap that remains in the fossil record does not make Archaeopteryx, or any other species, invalid as a transitional species. This is the fourth time I've had to make this point. LSS is once again pulling a Dr. Banjo. Furthermore, it is not like all scientists were just scratching their heads or ignoring this temporal paradox, which was coined by Alan Feduccia as part of his dogmatic birds are not dinosaurs position. Although the proposition that birds were the last surviving dinosaurs used to be controversial, especially in the 1990s, Feduccia's temporal paradox argument was criticized before 2010 and has become more untenable as we discovered more pre-Archaeopteryx Silorosaurs.
Back to the point, the authors list a handful of fossils and say that these discoveries provide significant new information on avian origins. What Jackson doesn't disclose is that this is a highly controversial statement in paleontology. Avian evolution is fraught with phylogenetic uncertainty. It's a can of worms. Some authors consider these to be as bird-like or more bird-like than Archaeopteryx, while others consider the same taxa as less bird-like than even the Troodontidae and Deinonychosauridae. It is still a mess. Maybe I'll make a more in-depth video on it later, but the temporal paradox problems with bird evolution are hardly solved.
That was a lot of hand-waving. Some authors consider these as bird-like or more bird-like than Archaeopteryx, while some consider some to be less bird-like than Troodontidae and Deinonychosauridae. Some people think weasel words are great. As he said himself, LSS doesn't bother to back up this statement with the details, so there isn't a lot to respond to here. But I will give him some courtesy and make the point for him. The Zhu et al. Paper from 2010 lists 5 species that fill the gap of pre-Archaeopteryx Silorosaurs. Guanlong, a basal tyrannosauroid, Haplachyrus, a basal alvarezsauroid, Anchiornis, a basal troodontid, and finally 2 species, Epidexipteryx and Epidendrosaurus, aka Scansoriopteryx, of the family Scansoriopterigidae, which is placed as sister group to Archaeopteryx and the rest of Avialae. That's how this paper classified these 5 species in 2010. Have these classifications been disputed by other authors? Well, Guanlong's placement as a basal tyrannosauroid and Haplachyrus's placement as a basal alvarezsauroid remain uncontested. Regarding the Scansoriopterigidae, their precise affinity was very controversial at the beginning of the early 2000s, but it has been narrowed down a bit. Scansoriopterigids were placed as sister to Avialans by some authors. Others suggest that they are sister to Oviraptorosaurs. Some did suggest that they are closer to modern birds than Archaeopteryx. However, most studies favor a more basal position, not closer to birds than Archaeopteryx is, so the general agreement is that Scansoriopterigidae lies somewhere within Pennaraptora, but outside Avialae. Lastly is Anchiornis. It has been considered by other authors to be the next most basal avialan after Archaeopteryx, which was also proposed in a very recent study by Behe et al. Other authors have also argued that it is very closely related to Archaeopteryx, but part of the same family and placed among Deinonychosauria. However, most authors don't consider Archaeopteryx as a Deinonychosaur and place Anchiornis as a basal troodontid, just like the Zhu et al. Paper from 2010. The general consensus is that Anchiornis is placed among the Paraves. Let's look back at what LSS said about these species. Some authors consider these to be as bird-like or more bird-like than Archaeopteryx, while others consider the same taxa as less bird-like than even Troodontidae and Deinonychosauria. That's definitely not true for Guanlong and Haplachyrus, and while Anchiornis is considered by some authors to be as bird-like as Archaeopteryx and Scansoriopteryx as more bird-like, the majority do not. The reality is very different from the misleading impression that LSS gives. It's true that determining precise phylogenies can be difficult, but this doesn't invalidate their general classification, such as the classification of birds as theropod dinosaurs. That has not been controversial among paleontologists for over a decade, much to the dismay of bandits like Feduccia. Nor does phylogenetic uncertainty of some taxa invalidate transitional fossils. Quite the opposite, actually. This is another consequence of transitional fossils. As was noted before when discussing whale evolution, the more transitional fossils, the more they blur the lines between groups. Consequently, determining the precise phylogeny of all these species becomes more challenging since the distinctions between different groups become increasingly more subtle and difficult to define, such as the difference between birds and dinosaurs. As the paper, An Overview of Non-Avian Theropod Discoveries and Classification says, quote, "The anatomical distinctions between non-avian and avian theropods are, therefore, particularly subtle and vary according to the phylogenetic analysis performed by authors." Close quote. Before, we asked LSS how he would define the differences between whales and other mammals. Now we can ask the same question regarding bird evolution. What are the differences between birds and dinosaurs? Where does LSS draw the line here? Before we move on, one final thing needs to be pointed out. Remember that LSS originally claimed that the temporal paradox, formed by a lack of Silorosaurs before Archaeopteryx, hasn't been solved, i.e. he is claiming there are no pre-Archaeopteryx Silorosaurs. However, I provided a paper demonstrating that pre-Archaeopteryx Silorosaurs have in fact been found. But what does LSS do after receiving this information? He pivots from his original claim and begins to weasel and hand-wave about the phylogenetic uncertainty of these species, while avoiding the fact that they are still pre-Archaeopteryx Silorosaurs that resolve this temporal paradox, regardless of the uncertainty about the precise placement of some of them.
Okay, what about Tectalic? They say that Tectalic was not dethroned as a transitional fossil because it was never considered a direct ancestor of tetrapods, even before these trackways were discovered. All the footprints showed was that the tetrapods or fishopods diverged much earlier than was previously evident. It's great that the initial paper was cautious in its claims for Tectalic and that scientists are willing to update things as more data comes in. This is what science is supposed to do, but it seems like Jackson is unaware of how Tectalic was presented and the importance placed on it by other prominent scientists. For just one example, Tectalic was such a huge deal precisely because it was supposed to be a prediction, when and where it was found stratigraphically, that was marvelously fulfilled in a stunning vindication of the evolutionary theory's predictive power, the most tangible evidence that evolution is true. It could have actually been a direct link in a true transitional form, and that it actually might have been your distant ancestor. One of the greatest fulfilled predictions of evolutionary biology. It was not only anticipated but predicted to occur in rocks of a certain age and in a certain place. Marvelous. But as it turns out, this is a failed prediction.
Again, notice how LSS has pivoted away from his original claim that Tectalic was dethroned as a transitional fossil and has now moved on to a different claim. The prediction that led to the discovery of Tectalic failed because it was based on faulty premises. Even if true, Tectalic's status as a transitional species remains uncontested. So it appears that when his previous arguments are shot down, LSS simply moves the goalpost to a different point, which is ironic since LSS later continues quoting Luskin, accusing scientists of doing the same thing with Tectalic. Anyway, LSS is making the case that since the tetrapod footprints predate Tectalic, then Tectalic was actually a failed prediction. This isn't really accurate. The prediction that led Neil Shubin and colleagues to spend months in the Canadian Arctic over several field seasons was that a transitional form between lobe-finned fish and tetrapods should be found prior to the fossils of the earliest known tetrapods, which back in the early 2000s was about 365 million years ago. They also narrowed their search to rocks younger than ones that contained some lobe-finned fish that already had some tetrapod-like features, such as Misunderstanding and Pennaraptoran, which are about 385 million years old. That doesn't mean they were discounting the idea that any transitional forms could be found before, or even after, this time period. This led to the discovery of a transitional form between lobe-finned fish and the earliest tetrapods, Tectalic, dating to about 380 million years ago. So the prediction was successful. They found exactly what they were looking for, a transitional fishapod that's between 365 and 385 million years old. Now, we know that the earliest tetrapods existed as early as 390 million years ago, not 365 million years ago. So if we were planning the same kind of fossil hunting expedition to look for transitional forms, we'd have to go looking in rocks older than 390 million years old, right? Well, only if we were looking for individuals or species that might have been ancestral to all tetrapods. As we discussed in the previous rebuttal videos, nothing about evolution says that after a group evolves, all its cousins must go extinct. There's no reason to think that these cousin species wouldn't persist later than 390 million years ago, up to, say, 380 million years ago. Personally, I've never been a fan of the way Tectalic was hyped as a successful prediction of evolution. Not because it wasn't a success, but because it implied that Tectalic is some kind of unique example of a prediction of evolutionary theory. In reality, predicting what kinds of fossils will be found in rocks of a particular age and environment is what all modern paleontologists do on a day-to-day basis. It's how all new fossil expeditions are planned. With limited time and budgets, paleontologists can't just go out and start digging willy-nilly. A paleontologist who's a specialist in early bird evolution wants to find early bird fossils. So to narrow down their search, they can, based on their knowledge of evolution, immediately ignore all the rocks prior to the Jurassic, for example. Precise details of timelines can be flexible, as illustrated by this tetrapod example, but especially on the large scale, they tend to be pretty firmly secure. As time has gone on, the trend is towards tightening up our understanding of precise timelines, allowing more precise predictions. Meanwhile, ID proponents and creationists sit silently on the sidelines, only piping up to jeer occasionally when something unexpected for evolutionists comes along, no matter how short-lived the confusion lasts. Now that the search is on for earlier tetrapod transitional forms, how will they react in the coming years when these fossils are inevitably found, filling the chronological gap? I suspect they'll react in much the same way they do today to the excellent morphological transitional series, with quiet grumbling and weak hand-waving. The discovery of the fossil footprints that proves tetrapods lived prior to the specimens of Tectalic that we found dethrone these individuals as ancestral to all tetrapods, but they remain transitional forms.
Finally, let's look at the Durant and Schmidt calculations. PZ claims that Michael Behe is a very bad mathematician. It's true that the Duretschmidt paper was aimed at showing that Behe was wrong. I'm really glad that this was brought up. Yes, the Duretschmidt paper attempted to disprove a model presented in Behe and Snooke's previous work. But ironically, they ended up strongly reconfirming the central thesis. The standard math of population genetics establishes the implausibility of Darwinian mechanism to produce what it needs to produce with the time and population resources that are available to it.
LSS says the 2008 Durant and Schmidt paper was aimed to disprove a model presented in the 2004 Behe and Snooke paper. This already shows that LSS has not read the paper in question, nor even the responses written by Behe himself to Durant and Schmidt. If he did, he hasn't read them very carefully. This wasn't the goal of Durant and Schmidt's paper at all. They only mentioned the Behe and Snook paper as a side note, dedicating just 4 sentences to it at the end of the paper, pointing out a problem they observed in addition to those pointed out by Michael Lynch. Durant and Schmidt's main criticism was outlined earlier in the paper, where they applied their calculations to an argument Behe made in his book, The Edge of Evolution, from 2007. Behe's argument concerns the mutations that made the malaria pathogen resistant to a drug called chloroquine. Oh my, how topical. In the early 2000s, it was shown that the resistance was the result of 2 mutations in the PFCRT transporter protein. A single mutation doesn't confer resistance. Both were required. Behe claims that both mutations had to occur simultaneously, and that the probability of this event is the odds of a single mutation, 1 in 10 to the power of 10 squared, which is 1 in 10 to the power of 20. For something like this to happen in humans, Behe says that it would take on average 100 million times 10 million years, or 1,000 trillion years. Durant and Schmidt tore into this argument, not the Behe and Snook paper in particular. Durant and Schmidt originally calculated that the actual value would be 216 million years, a difference with a factor of about 5 million. So LSS is already off the mark here.
Behe himself had to write a rebuttal in which he stated that the Durant and Schmidt paper was, quote, seriously flawed, unquote. So it's seriously flawed when it annoys Michael Behe, but the Discovery Institute is happy to gloss over that part to cherry-pick conclusions they like from this seriously flawed paper.
No, data was not cherry-picked. Their entire conclusion, as mistaken as it was granted. Behe called it seriously flawed because it contained serious flaws. Durant and Schmidt themselves acknowledged as much in their reply to Behe.
Durant and Schmidt originally calculated that Behe overestimated his number by a factor of 5 million. They acknowledged a correction by Behe which divides this overestimation by 30. That is indeed a significant difference, although even with this correction in mind, it would mean that Behe's number of 1,000 trillion is still overestimated by a factor of about 150,000.
First, correcting for a simple oversight of using the incorrect mutation rate, then correcting for their neutral rather than deleterious model, because again, the empirical data suggests the intermediate mutations are not neutral or harmless, and adjusting for the effective rate of the second mutation and their inappropriate multiplication of probabilistic resources, their calculations were off by more than 7 orders of magnitude. When correcting for these errors and others, their model agrees quite well with the original paper by Behe and Snook, as well as the empirical public health data and the literature.
The first point we already covered, but LSS here highlights 3 more of Behe's objections to Durant and Schmidt. First objection is that the first mutation should be taken to be deleterious instead of neutral. Secondly, that the effective rate of the second mutation was omitted. And third, the inappropriate multiplication of probabilistic resources. LSS claims that these mistakes cause their calculations to be off by more than 7 orders of magnitude. However, the third point was not even a relevant factor to Durant and Schmidt's calculations. When Behe took Durant and Schmidt's numbers for granted, he noted that their 216 million year waiting time for humans is still prohibitively long. Durant and Schmidt pointed out that their calculations only consider a specific pre-specified pair of mutations in one gene. They noted that humans have thousands of genes where similar beneficial mutations could take place, which would decrease the waiting time even further. It's basically the same thing we had to explain to LSS. The difference between one specific person winning the lottery and someone winning the lottery. Of course, Behe disagrees with this since he describes this as committing the gratuitous, inappropriate multiplication of probabilistic resources. I will expand on this point shortly, but it first must be noted that this had nothing to do with Durant and Schmidt's original number as LSS mistakenly insinuates. Now the second point, where Behe argues that Durant and Schmidt forgot to take into account the success rate for the second mutation. For this, you need to know a little background. Durant and Schmidt calculated the probability for 2 mutations to occur, which only together would confer a beneficial phenotype. The second mutation in particular would complete a transcription factor, or TF, binding site, which is almost complete. Like, 9 out of 10 nucleotides are good, but the last mutation must occur for it to complete 10 out of 10. Behe points out that waiting for a mutation to come along that produces the TF binding site, there was a higher chance that another mutation would cause this 9 out of 10 to become an 8 out of 10 incomplete binding site. However, Durrett and Schmidt rebutted by pointing out that this is only relevant when you assume that there is only one individual with the first mutation. If you have a large set of individuals with the first mutation and the 9 out of 10 incomplete TF binding site, then when a mutation occurs in one individual that causes it to have an 8 out of 10 incomplete TF binding site, there are still many other individuals remaining who have the 9 out of 10 TF binding site. Behe doesn't understand why this is relevant. He says that in his letter, he, quote, wrote no such thing as there being only one individual, close quote. But that doesn't matter whether Behe explicitly assumed there was only one individual. The fact is that his objection would only count if there is only one individual with the first mutation. The first objection is about whether the first mutation is neutral or deleterious. This is the big difference between their models. Behe thinks the first mutation is very likely deleterious, which is why Behe assumes that the two mutations had to have happened at the same time in one individual in order for chloroquine resistance to have evolved in the malaria pathogen. Durant and Schmidt, however, modeled the first mutation to be neutral, which means that the first mutation could have spread throughout the population by drift alone, long before the second mutation occurs. But Behe points out that if you assume the first one to be deleterious, which he thinks it likely is, then natural selection would make it unlikely for the first mutation to have spread through the population. Though LSS highlights this particular section while claiming that the empirical data suggests that the intermediate mutations are not neutral or harmless, he gives no references to this empirical data, and neither does Behe in the reply LSS highlights. However, on other occasions, Behe likes to cite one paper from 2005, which shows one of the mutations involved is indeed deleterious. However, it also shows that the variants possessing all the other mutations, except for K76T, aren't deleterious. As the paper reads, quote, these results suggest reduced parasite viability resulting from K76T in the absence of other PFCRT mutations. This situation is not reciprocal, however, in that parasites harboring all the other mutations except for K76T, illustrated by our back mutants, show no signs of reduced viability in culture, close quote. So this particular mutation, while detrimental in and of itself, could occur as the last mutation needed to confer resistance, thereby having a net positive contribution on fitness in the presence of the drug. And lastly, the paper Combinatorial Genetic Modeling of PFCRT-Mediated Drug Resistance Evolution in Plasmodium falciparum from 2016 shows that resistance can be achieved by an evolutionary route of 4 mutations with increased fitness after every single mutation along the way. So even a single mutation can confer a slight increase in resistance. Not only that, it shows that there are multiple plausible routes for the evolution of drug resistance in malaria. And finally, the paper also highlights the importance of modeling different selective pressures in order to understand how evolvable particular genotypes are, instead of looking at the fitness effect of a mutation under 1 or 2 environments. It's often a lot more complex than that.
Next, they accuse Durrett and Schmidt of calculating the waiting time far too narrowly. They don't model how evolution actually works.
I didn't accuse Durrett and Schmidt of calculating the waiting time far too narrowly. P.Z. Myers did say that the conditions laid out by Durrett and Schmidt were very narrow and don't represent how evolution actually works. And he is right in that regard, since evolution doesn't just operate within those narrow parameters. Even Durrett and Schmidt said the same thing in their response to Behe, as was noted previously. However, just as P.Z. said, given those limited parameters, you can't argue with their math, so their numbers are totally fine, as long as you keep in mind that they're only valid under the narrow conditions that Durrett and Schmidt defined, and this is exactly where Behe made a mistake. He claimed that Durrett and Schmidt calculated how long it would take for organisms to evolve 2 simple beneficial mutations. However, it was actually about 2 pre-specified mutations at specific loci, not any 2 beneficial mutations. There is a significant difference between these two, which I have to explain again to LSS after the next clip. Furthermore, as I pointed out in the first response video, LSS mistakenly claimed that the numbers a few million years for fruit flies and 200 million years for humans were about 2 beneficial mutations, while they were in fact about 2 neutral mutations for fruit flies, and 1 neutral and 1 beneficial mutation for humans. LSS has not responded to or acknowledged these corrections.
Because humans and fruit flies have lots of places in the genome where beneficial mutations can take place, which increases the odds substantially and shortens the waiting time as a result. But no, in fact, Durant and Schmidt are right in how they modeled it. This is the proper way to estimate the power of Darwin's mechanism, because almost all possible single or double or triple mutations amongst sequence space will be useless. It would be silly to think that just any 2 mutations in general would be adaptive. The vast majority are completely ineffective or degradative. This is why they chose to model it so specifically. So this is not like a specific individual winning the lottery. That's a gross misunderstanding of the model.
LSS says that this is the reason why Durrett and Schmidt chose to model it so specifically. Again, Durrett and Schmidt's goal was to address Behe's calculations regarding 2 pre-specified mutations. So to keep it relevant to Behe's claim, they kept it at 2 pre-specified mutations. That's the reason why they modeled it so specifically. Not for the reason LSS claims. Moving on, just like Durrett and Schmidt explained to Behe, I pointed out to LSS in the previous video that there is a difference between the odds of any 2 beneficial mutations and the odds of 2 beneficial pre-specified mutations. The difference being that the genome contains lots of other loci besides the 2 pre-specified ones where beneficial mutations could take place. LSS objects to this, saying that, quote, almost all possible mutations will be useless. It would be silly to think that just any 2 mutations in general would be adaptive. The vast majority are completely ineffective or degradative, close quote. First, I couldn't help but notice that these are the exact same lines word for word that Behe used in his reply to Durrett and Schmidt. Yet, LSS doesn't make it clear that he is directly quoting from Behe, implying that it is his original commentary. Plagiarism isn't a good look. Anyway, did anyone else catch the major blunder? Let me explain using the lottery analogy again. LSS claimed that Durrett and Schmidt calculated the odds of someone winning the lottery. I pointed out that actually, no, they calculated the odds of a specific person winning the lottery. The odds of someone winning and the odds of one specific person winning are not the same because there are more potential winners beside the one specific person. To adapt his response into this analogy, LSS objected by saying that almost all humans on Earth do not participate in the same lottery, so it would be silly to think that just any person on the planet could win the lottery. Can you now see who is actually grossly misunderstanding the other? I did not claim that every human would participate in the same lottery. I pointed out that there are more than just one person playing the lottery. I agree that the vast majority of all humans do not participate in the same lottery, but the number of participants can still be far more than just one individual. Hence, the odds of someone winning is still far greater than the odds of a specific person winning. Now the explanation without the analogy. I did not claim that any possible mutation in the genome would be beneficial. That's a gross straw man of my position. I pointed out that there are more places in the genome besides only 2 loci where beneficial mutations could take place. I agree that the vast majority of all possible mutations are not beneficial, but that doesn't mean the number of all possible mutations that are beneficial is anywhere close to only 2 specific mutations. The number is far higher, even when you narrow the focus to beneficial mutations regarding a specific beneficial phenotype, like chloroquine resistance that was noted previously. Hence, the odds of 2 beneficial mutations is still far greater than the odds of 2 pre-specified beneficial mutations. Do you get it now, LSS?
And finally, a 2005 paper that purports to shred Behe and Snokes' calculations. This one is pretty simple. The authors wrote a reply answering Lynch's paper, and in it, Behe and Snoke write, our model posited necessary intermediate mutations to be deleterious in the unduplicated gene. Lynch's model assumes them to be neutral. All of his objections to our work stem from this difference. Experimental studies contradict Lynch's assumption of complete neutrality as a rule. The majority of amino acid substitutions decrease protein function.
So, as LSS noted, the main difference between these papers is that Lynch assumed the intermediate mutations to be neutral, while Behe and Snoke assumed them to be deleterious. Lynch already refuted that in his paper. Quote, second, Behe and Snoke assume that all mutational changes contributing to the origin of a new multi-residue function must arise after the duplication process. They justify this assumption by stating that the majority of non-neutral point mutations to a gene yield a non-functional protein. To stretch this statement to imply that all amino acid changes lead to non-functionalization is a gross mischaracterization of one of the major conclusions from studies on protein biology. Most protein-coding genes are tolerant of a broad spectrum of amino acid substitutions, close quote. And, quote, thus most proteins in all organisms harbor tens to hundreds of amino acid sites available for evolutionary modification prior to gene duplication, close quote. He points out that, according to most studies, even the ones that Behe and Snoke cited, protein-coding genes are rather tolerant to amino acid changes. This is known as protein robustness, and has been known well for a long time. This was especially shown by the technique called alanine scanning, which was developed by James Wells in the late 1980s. It involves making many mutant versions of a protein where one amino acid is replaced by alanine, the simplest amino acid after glycine, in order to determine how this would affect the protein. Here is a more extreme version of alanine scanning, where instead of replacing each site with alanine, they replace each site with all of the 20 amino acids, of course one at a time. This produces a mutation sensitivity map. The color yellow denotes that the particular substitution had little influence on the stability of this protein. Only at the dark places shows which mutations have a strong detrimental effect. This shows that, for the most part, proteins contain a relatively small number of residues that are really important to their stability and functions, whereas for the others there is a lot of leeway, meaning amino acid substitutions in those places are mostly tolerated.
That's basically it. There were other minor objections that Jackson and PZ cover, but these are the main objections. As I wrap up, I just want to say thank you to them for engaging in the discussion. I know it takes a lot of time and effort to create videos like this, and this sort of open discussion lets people see for themselves the strengths and weaknesses of both sides.
I couldn't agree more. So, thanks for watching, and I will see you all next time.